Welcome, ladies and gentlemen. Thank you for coming tonight, especially given that the San Francisco Giants are playing. I'm really glad everyone could come. I want to thank also University of San Francisco for its commitment to a superb education. And I would like to thank the McLaren Scholars, and in particular Alex Spady, for all their help, and also Dale Smith for all her help. We have a lot of exciting speakers today. They're all professors at the University of San Francisco, and they're going to spend about 10 minutes speaking about the Nobel Prize in their area. And then, if you could, just hold your questions till the very end. And at the end of all speakers, we'll open it up for Q&A. Okay, the first prize that we're going to discuss is economics. And the professor speaking about this is Suparna Chakraborty. She's in the economics department. She obtained her PhD from Minnesota in economics. She currently teaches international trade and economics and macroeconomics at USF. Her research is really exciting. It's in international finance and financial crises, and she's written papers such as Foreign Direct Investment and Regulatory Remedies for Banking Crises. So please welcome Suparna Chakraborty.
All right, can you guys hear me? All right, okay, so the Nobel Prize for Economics, it's kind of interesting because this was not something that the Nobel Committee had in mind before. So this is not one of the original disciplines in which a Nobel was offered. So let's see a little bit of the history of it. So this was never intended. So Nobel Prize in Economics is the newest prize, Nobel in economics, and it was really established in 1968, right? So when we started thinking about banks and we started taking banks and the financial system seriously, that's when people thought, you know what, economics is also a subject, so let's give them a Nobel, right? And the fund for the Nobel Prize in Economics comes from the Central Bank of Sweden, right? So that's how we had the first Nobel, and the first prize was awarded in the year 1969, okay? And it was given to Ragnar Frisch and to Jan Tinbergen. So they were the first recipients of the Nobel Prize in Economics. So let's just fast forward to the present time. It's 2012. The prize was just announced a few days ago. You might have seen it in the news. And this year, the prize has been given to two game theorists. The first one is Professor Alvin Roth, and the second one is Professor Lloyd Shapley. Okay? So these two gentlemen are the newest recipients of the Economics Nobel Prize. So just to give you a little bit more about what they do, where they are, and what their work is all about. So Professor Alvin Roth, he is currently at Harvard, and there is a good news for all of us here, and that is he will be moving to Stanford from January 2013. So he is actually currently a visiting professor at Stanford, okay? And Stanford was very lucky. They kind of made him an offer, I guess, which he couldn't refuse. So he will be joining us on the West Coast. So that would be great. So what does he work on? So he's primarily a game theorist, okay? And he also does a field of economics, which is called mechanism design. And I just wanted to say one thing, otherwise I'll feel very disloyal to Minnesota. And that is the last year's Nobel Prize in Economics was awarded to Professor Leonid Horowitz, who was is called the father of mechanism design. Okay, and I was very lucky to be in one of his classes. He was 90 years old and he was sharper than we are. Okay, so he is the oldest recipient of the Nobel Prize in Economics, in fact, all Nobel Prizes, and he was the father of mechanism design. And the tradition seems to be continuing even this year. So Alvin Roth, he works in mechanism design. Now, for many of you who don't know what mechanism design is or what we mean by game theory, what does he do? So Alvin Roth is actually famous for his work on matching. So what does he really do? He has written some very clever algorithms which are applied to health sciences. So basically, he has something which is called the National Resident Matching Program. And this has been used in New England, and it has also been adopted all over the United States now. And if you are in the medical field, you might know someone who is studying medicine. When they become residents, they have to be matched up with their best placement, right? The algorithm that Professor Roth created is designed to match the residents with the best placement. And it is widely used in medical field nowadays, okay? And he has kind of continued his work in this confluence of, we economists are kind of a funny bunch. We work in all areas, okay? So he has kind of diverted his interest a little bit towards health. And currently what he is working on is a very interesting program which has been adopted in New England. And it is called the New England Program for Kidney Exchange. So if you are waiting for a kidney transplant and there are kidney donors out there, right? His algorithm is a quick way of matching the donor with the recipient, okay? So this is what he has been working on. So there's a lot of practical applications that we are getting from Alvin's work. And it's really going to be wonderful to have him here at Stanford. Hopefully, we at USF can make him come down here and give us a talk, right? So that would be really great. The second person who actually jointly received the Nobel Prize, I think there were about 72, prizes awarded, and 44 of them are joint Nobels. So the second person who won the prize along with Professor Roth is Professor Lloyd Shapley. Okay, so Professor Lloyd Stowell Shapley. He is currently at UCLA. So West Coast won the Nobel Prize in Economics this year. Okay, so he's an emeritus professor at UCLA. He is also a game theorist, right? And for any of you who are interested in business economics or in financial economics, the first thing that you will ever hear in economics, financial economics, is a term called Shapley value, okay? And that Shapley value, Shapley in that Shapley value is Professor Shapley, okay? And what it is really, there is a strategy in game theory where we call, voters and the power of a voter. So if you are voting and your wife is voting or your girlfriend is voting, who wins, right? So that's the kind of studies that Professor Shapley has been doing. And he has created something called the Shapley value. And recently, so he is one of the oldest practicing economists in game theory. And since the 80s, he has moved away from his traditional theoretical economics to really studying power, bargaining. And I think it's very, very interesting that right now that we are midst of the election year, Professor Shapley won the Nobel Prize because he really studies bargaining and power plays, okay? And the reason why he won the Nobel Prize, amongst other things, is he created something called the Shapley-Shubik Power Index with Professor Martin Shubik. And this really is a way, an economic way of measuring how powerful you are if you're playing a game. Okay? So we have 2 Nobel laureates this year, both from the West Coast. I think we can claim Professor Alvin Roth and Professor Lloyd Shapley. So hopefully we will at least have one of them come here to USF to give us a talk on economics sometime very soon. So thank you for being here, and it would be wonderful if any of you in your later stages of life decide to major in economics, get a degree in economics, and maybe bring USF or economics, first prize, Nobel. How about that, right? Thank you guys for being here.
Thank you very much, Suparna. By the way, I did talk to Alvin Roth, and I'm trying to get him to come next year. So, but probably a year before we'll see him because they're paying him mucho dollary to go to other places. That's what happens when you win the Nobel. Thank you very much. Our next speaker is in medicine, and it's James Sykes of the biology department. He obtained his PhD from the University of Maryland. He did his postdoctoral work at Howard Hughes Medical Institute and the University of Illinois at Urbana-Champaign. He currently teaches developmental biology, evolution, and invertebrate zoology. Wow, I think I should take his course. His research is in the field of regeneration biology and the role of stem cells in post-embryonic developmental processes. Wow. No, seriously. Please give a warm welcome for James Sykes.
All right, thank you guys for coming. Today I'm going to tell you guys a little bit about the Nobel Prize in Medicine or Physiology. So the prize this year deals with stem cells, so it's something that's dear and close to my heart. So the prize this year is a shared prize, as Naparna actually mentioned earlier. It's shared by these two professors, Dr. Sir John Gurdon, who is a professor at the Gurdon Institute that he actually created, and Shinya Yamanaka, who is actually a professor at Kyoto University, but spends most of his time here in San Francisco at the Gladstone Institute. So these two guys both did foundational work that has led to a rather dramatic finding in stem cell biology that has basically changed the way we think of stem cells today. So if we think about stem cells and overall developmental biology that occurs not only in humans but all animals, is that we have cells that begin as a sperm or egg, as a zygote, very undifferentiated, can become anything, and through time, these pluripotent cells go on to become our liver, our heart, our skin, and different things like that. So we have this idea that we can actually harvest these cells, okay, and actually use them to actually, in stem cell therapies, and you guys have all heard of embryonic stem cells. So embryonic stem cells are these pluripotent cells that are found in this part of a developing mammalian embryo. And from these cells, normally we go on to develop an entire human, an entire mouse, an entire frog, whatever we're looking at. And during this process, some of these cells, these embryonic stem cells, go on to choose a path and either become a neuron, a nerve cell, a muscle cell, a brain cell, or a different kind of cell like that. The thought has always been that once a cell has decided what it's going to become, it never changes its path. So once a nerve cell thinks it's a nerve cell, it's going to stay that for the rest of its life and never go back to an undifferentiated state. So what this Professor Gordon did back in 1962 is he worked with this guy. Isn't he beautiful? So this is a frog that lives in Africa. It's called Xenopus. It's the genus of this frog. So he worked with these frogs. And what he did is he took a frog eggs, because frog eggs are very easy to manipulate and to obtain. So he took a frog egg and simply irradiated it. He put UV light on it and basically killed the DNA. So he killed the nucleus in that frog egg. He then took intestinal cells from a Xenopus tadpole, so one of these frog tadpoles, took the nucleus from this cell and transplanted it into the egg that had no nucleus. And lo and behold, what occurred is the cell actually went on to develop an entire frog. So he showed for the very first time back in 1962 that a nucleus of a differentiated cell can gain that ability to make every cell in the body. Okay? Even though it had the genetic program for only intestine, it actually made neurons, it made skin, it made the entire embryo. The embryo, which grew up to become an entirely adult frog. So it kind of changed our whole thinking of how maybe this DNA inside of the nucleus may not be differentiated for life. It may have the capacity to change. Since his discovery in 1962, we've all heard about Dolly. Dolly was one of the first mammals that was cloned, and we've actually cloned a variety of different animals through this technique. So he basically he led the foundations for all of the cloning that we think about today. So basically, his discovery led us to believe that a differentiated cell nucleus has the potential to go back and become what we call pluripotent, the ability to actually change into a different type of cell. But a major question still arises, and it's the fact that if this one cell can actually gain its fate, can we take these same cells, not necessarily just the nucleus, but can we take that cell and reverse it back to that immature state? Can we reverse these arrows in a single cell from the body? And this is where Dr. Yamanaka's discovery comes into play. He started working in the mid-2000s, okay, in the last several years, and he realized his work was dedicated in using mice and looking at their embryonic cells, okay, and trying to figure out if we can actually reverse these processes. So his first goal was to look at these embryonic stem cells and identify which of the genes were specifically expressed in those embryonic stem cells. And he identified 24 of these genes that were differently expressed in embryonic stem cells here versus all of the other types of cells in the body. So basically he whittled down from these 24 to realize that there were actually only 4 genes that are truly different and that actually allow a cell to become pluripotent. And these are the cells, these are the genes, and as you can see, genes have funky little abbreviations. But so these were the 4 genes that he actually realized were the genes that actually led to pluripotency. So what did he do? He took these 4 genes, okay, identified here as a test tube. He took these 4 genes and implanted those genes and turned those genes on in a cell, in a skin cell from a mouse. Okay? And then what happens is that one cell, lo and behold, actually became an undifferentiated cell. So we took a fully differentiated cell and actually made a stem cell out of it that, when implanted into another mother mouse, grew up to become a new mouse itself. So for the first time now, we have opportunities to take differentiated cells, introduce just a couple of genes, 3 or 4 genes, and it makes them a stem cell, very similar to the embryonic stem cells that we actually study. So he named these cells induced pluripotent stem cells, or iPS cells. Okay? Basically, the impacts of these discoveries have changed the entire field of stem cell biology and even medical biology at this point. So because of these observations and the fact that very few transcription factors, very few genes are leading to the undifferentiated state from a differentiated cell, we have the potential to look at transdifferentiation, taking one type of cell from our body, allowing that to change into a different type of cell. And then obviously we can also look at disease research, looking at taking cells that are diseased, taking them back into an embryonic state, okay, or undifferentiated state, trying to figure out what's different about those cells. Okay? So again, taking any cell, and then we can study basically a disease in a dish, is what they call it, but taking these cells, growing them up, and looking at their disease state, trying to figure those things out. And finally, this work has also removed the need to use live human embryos to harvest stem cells. We all heard the ethical debates about the use of embryonic stem cells in biological research. Very few people actually use embryonic stem cells anymore in biology because we have this capacity now to induce stem cells from any type of cell in the body. So it's actually revolutionized the ability and even minimized the ethical dilemma that we actually have by harvesting these inner cell mass cells from an embryo, and allows us to now create our own stem cells that can be used for medical research as well as pure research itself. So again, thanks to the work of these 2 guys, we now understand that a differentiated cell, your skin cell, still has the capability to go back and become a pluripotent cell that can become anything in your body. Has completely changed the paradigm of what we thought was the case in biology. So with that, I just wanted to give you guys a quick overview of what these 2 guys did. One using a frog back in the 60s, and one in the last 4 or 5 years using mice and using stem cells for mice. So with that, I'll let you guys ask questions at the end, and I'll pass it on.
Wow, that was exciting, huh? So I learned something there. Our next speaker is in another interesting field, which is physics. And this is Seth Foreman, who's in the astronomy and physics department. He obtained his PhD at JILA in Boulder, Colorado, where, curiously enough, Dave Wineland, a Nobel recipient, comes from. He also did postdoc at Stanford University, and he currently teaches calculus-based physics for science majors and an upper division mechanics course. Suggest if you haven't taken it, you might want to take it in the next couple years. His research involves working with lasers and optical clocks. Would you please give a warm welcome for Seth Foreman?
All right, thanks for coming out, especially for the few students in my class who made it here tonight. It's good to see that so much of University of San Francisco are Giants fans. I applaud that, but I'm glad that you all were able to make it here too. Okay, without further ado, here's the 2012 prize in physics. It's these two gentlemen, Dave Wineland, right here, this nice, rugged, Rocky Mountain type looking fellow, and a Frenchman, Serge Haroche, who's looking, in my opinion, quite French as well. So I think that the photos do them justice. Dave Wineland works out of NIST and CU Boulder, working at Agila in Boulder, Colorado. Serge Haroche is right now affiliated with the Collège de France in Paris. And so I just wanted you to see their friendly faces before we talk about what they did. So I'll tell you the what. What did they do first? According to the Nobel Committee, if this will work, there we go. The Nobel Committee said that they had groundbreaking experimental methods that enabled measuring and manipulation of individual quantum systems. So that sounds like technical jargon because it is technical jargon. So what it really means in English is that they spent several decades, 30 years is really just a placeholder here, of solid work figuring out how to directly observe and interact with some of the most fundamental processes in quantum mechanics. So I want to tell you a little bit, just a little, don't worry, don't get scared, about quantum mechanics. But in my opinion, there's another reason that at least one of them won the Nobel Prize. And I think that you've already kind of taken a good guess at it. Here it is. On the Nobel website, someone made the comment, "Congratulations, Dave, you were always going to win it with that mustache." So this is the face of a physicist. I aspire to that someday. Okay, so what? We're going to talk about superposition and decoherence, and those are also technical jargon, so let me talk about what those mean first. First, let's talk about what a quantum system is. A quantum system is not as fancy as it sounds. It can be something as simple as a single atom that's interacting with light. And in general, a quantum system, a cheeky answer might be that everything is a quantum system. Some physicists might tell you that the entire world works at its fundamental level based on the principle of quantum mechanics. And it's a little seductive to want to believe that. So far, no experiment has ever contradicted the laws of quantum mechanics that we know of. But there are also other problems with it that if you want to learn more about, you can come talk to us physicists in the physics department about. So that's the cheeky answer. And the real answer is that a quantum system tends to be, the kinds that we're interested in, tend to be systems of very, very few, very tiny, very fundamental objects that as a result exhibit some rather strange behaviors that we're not used to. So the atom interacting with light, you might remember a cartoon picture of it might look something like this. If you took high school physics or chemistry, you learned that you had an electron orbiting around some nucleus that could be as simple as just a single proton. So this is one electron, one proton, and one photon. There's a three-body system. Okay, there are three things interacting with each other. It's light interacting with an atom. And this is a very, very quantum mechanical system. But a more appropriate picture for what it might, I shouldn't even say what it would look like, but one way to visualize it is that it actually is something like this. The electron is not orbiting some nucleus. The electron is smeared out over all space in a region that's about the size of what we call an atom. Okay? And the electron has a probability of being found anywhere in this cloud. Okay? So this is a probability cloud. Chemists might call it an orbital. Okay? Physicists would say that this is the wave function of the atom. And all it means is the electron could be here, could be here. The electron tends to be where the light places are in this figure and not where the dark places are in this figure. And these orbitals come in many shapes and sizes. Here are, for a hydrogen atom, some different shapes of where the electron might be found. And they correspond to the different energy levels. So if you remember that an electron orbiting the atom in your cartoon picture here can transition from a high energy to a low energy and give off some light, that might look like it changing from being found in these locations to being found in just these locations. And when it does that, it gives off some light. And so very naturally, what you see right away is this principle of superposition and decoherence. Superposition means, hey, the electron is not in one place at any given moment. It's really got a probability of being found in many, many places. And that should sound very strange and freaky to you because there's not usually a probability that you find me in more than one place at the same time, no matter how hard I try. So the electron doesn't have to work very hard to be in more than one place at the same time. And in fact, the electron can actually be in both energy levels at the same time in some sense. It can have a probability amplitude of being in either of these kinds of orbital configurations at the same time. So it might be in both at the same time in some sense of that word. All right. So where do these things happen? They happen everywhere. Our universe is full of these quantum systems. Here's a giant hydrogen cloud out in outer space. It's a molecular cloud. It's got lots of different kinds of these atomic systems in it. And every little atom is its own little quantum universe doing these things. Clouds like this actually give off light that is similar to laser light, except that it's not in the visible part of the spectrum. It can be in the microwave part of the spectrum. So instead of being called a laser, it's called a maser. And that coherent radiation can be detected. So these things happen all the time in nature. And looking up in the sky and looking at the wavelength of these radiations and the energies is actually one of the ways that quantum mechanics was really built up in the first place, doing spectroscopy on these kinds of systems. Okay, so the universe is full of these quantum systems. That's the take-home message from this slide. So what did these guys do? Let's see. Dave Wineland basically traps these atoms, just a few of them, in a tiny little trap. So he's got little electrodes that have various electric and RF fields applied to them. And he can trap small numbers, just a few ions at a time, in the very center of his vacuum chamber in the lab. And then he shines laser light at them and watches how the light interacts with the ions. The flip side of the coin is what Serge Haroche does. He does the exact inverse. He traps photons in between two very, very shiny mirrors. They're the shiniest mirrors known to man. They're made of superconducting niobium, so he cools it way down, and he has light bouncing around in here, and the light bounces around something like 10 to the 10 times. It goes a total distance of like 40,000 kilometers before the light gets absorbed by any given mirror. So it bounces around a whole lot. And then he takes and he puts atoms through this cavity and lets them interact with the light. So in one case, we've got ions interacting with light, and in another case, we've got trapped light interacting with atoms. Very, very symmetrical Nobel Prize this year. Okay, what does this look like in the laboratory? Well, Dave Wineland, this is what his laboratory looks like. It's mostly laser beams. Both of these guys would be mostly laser beams. So he's got tables full of lasers. And then the heart of the experiment really looks as simple as this. Just a little tiny bunch of electrodes, and he puts this inside of a chamber and cools it down to very, very cold temperatures, and he traps the ions right in the center of this trap. In the meantime, let's see here. That's an ion trap. In the meantime, Serge Haroche is taking these superconducting mirrors, and he puts them together. You can kind of see here's the two mirrors that are shown right up here, and there's a small gap between them where the atoms can get in, and he has the light, the microwave light in this case, bouncing around back and forth, interacting with the atoms. Both of them are studying a system that should sound a little familiar to you. It's called a Schrödinger cat state. So if you've heard, have any of you heard of Schrödinger's cat? Okay, so Schrödinger's cat is a somewhat paradoxical thing. The idea is Schrödinger, one of the fathers of quantum mechanics, said, hey, suppose we put a cat in a box. We have this absurd thing that a system can be in two states at the same time. So let's make one of these atoms be in the two states at the same time. And let's have a little sensor that decides if the atom decides to be in one of the states, we'll let the cat live. And if the atom is in the other state, we'll kill the cat. So, I don't know, you can be as creative as you like. Maybe a guillotine slices the cat's head off. Maybe it's a bottle of poison gets cracked open. Everyone likes, a different way to kill Schrödinger's cat. But the point is, the system is in two states at the same time, and that means that the cat must also be in two states at the same time, both alive and dead. And that should sound both pessimistic and optimistic to you, depending on what kind of person you are. Okay, I prefer the cat dead, for instance. For Serge Haroche, I also put up the French version of Schrödinger's cat. This is a French cat that is alive and a French cat that is dead. Okay, so it's very strange for an object to be in two states at the same time. How could something possibly be both alive and dead, or have a probability of being both at the same time? And this has been a source of argument over the years. And both of these physicists, in their work for this year's Nobel Prize, have been investigating states like this with the technology I showed you. And Serge Haroche in particular was actually able to watch, essentially, the cat choose whether to be alive or dead. He was able to watch how this superposition of states, being both alive and dead at the same time, can change and choose one or the other. That process is called decoherence. If you'd like to know more about it, you will have to take a lot of math courses. Okay, so we're talking about superposition and entanglement and decoherence, and I'm almost done battering you with this. I want to tell you when the work was done. I think the most succinct way to tell you when the work was done is to show a picture of this nice 1980s mustache. So this is Dave Vineland. This work was done over the last several decades. It really was a long-term set of experiments that made progress in very small steps all on the way. So that's when the work was done. And then finally, I want to tell you what it's good for. Physics is generally a very, very fundamental discipline, and we're trying to answer questions like, how does the universe work in the most fundamental way? And so both of these gentlemen really are seeing themselves as doing fundamental physics research, looking at this process of decoherence and how quantum mechanics works at its most basic level. So it's very not applied, and it's very fundamental. But if you absolutely insist on having some applications for this, then we can tell you that it's good for things like optical clocks. So Dave Vineland's research can also be used to create, using these exact same quantum states, clocks that are accurate to 17 digits. It's the most precise clock ever created. And these clocks work using the technology of watching this decoherence happen in some sense. And another area of application would be quantum computers. So again, in Dave Vineland's experiments, you can trap these ions, and you can manipulate their quantum state and have them communicate with each other and exchange quantum information, and try to build a computer that has the interesting property that the bits are not either zeros or ones. They can also be both 0 and 1 at the same time. And that leads to potentially the ability to do very, very fancy things that normal computers cannot do. Okay, so to summarize, I think we were also asked to tell you whether we thought this was a good prize or not. I think this is a pretty good prize, but I want to point out that as both recipients would be quick to tell you, the field of quantum optics has many, many people working in it, and they would both be very happy to share credit with a large number of people who have worked on this over the decades. And I also want to just highlight that this particular Nobel Prize, in my opinion, has it all. We've got the fundamental physics of the universe, superconducting niobium cavities, which is, let's face it, that's just fun to say. It's got Paris, lasers, dead cats, the shiniest mirrors ever created, more lasers, mustaches, the most accurate clock, and more precision lasers. So I'm a pretty big fan of this year's prize. Thank you.
Thank you. Thank you very much. Our next prize is the Peace Prize, and our next speaker is also, I think, why USF is such a unique and wonderful place. Our next speaker is James Hamby, and he is at the Lane Center for Catholic Studies and Social Thought. He's currently the Lo Schiavo Chair in Catholic Social Thought. His PhD is from Oxford University on Hegelian Metaphysics of Trinitarian Theology. He currently teaches theology at USF, and he is currently writing a book on the theological foundations of Catholic social teaching. Please welcome James Hamby.
Donald Rumsfeld once referred to old Europe, and so I want to begin with an apology. I'm not only old Europe, but I'm also old technology. I'm afraid I don't have any overheads or PowerPoint or anything like that. It might be of interest for some of you. I don't know if you've been to Europe or not, but if you've traveled there or you intend to travel there, I hope you'll enjoy the variety of cultures and all the rest of it. But one of the things that you might just take for granted is the way in which you can move between different nations without having to change your currency, with the exception of the UK, of course, which is where I'm from. I'm a British province Jesuit. Now, you think that's okay because you do that quite simply in America. You move between states. You don't have to change your currency. It's all the same. Reverse that. Imagine what it would be like if you were moving from California to Ohio and you had to change currency. Or as you moved around between the states, you also had to change currency. I think you'd probably find it pretty inconvenient and maybe even confusing. If you then think, what would it take to put all of that together so that you have a common currency? not just economically, but also politically. And what's at stake in terms of a currency? The whole question of different sovereignties. Then you begin to understand both the complexity of Europe, but also the extraordinary achievement of having a European Union. That's why I think this Nobel Peace Prize going to the European Union is itself a quite remarkable prize, and I think a very worthy one. It's not unique to give the prize to a collective. It's been done before. It's been done, the prize has been given to the United Nations. It's also been given to the Red Cross. It's also been given to other organizations like Médecins Sans Frontières. In the citation for the Nobel Peace Prize, this is what Alfred Nobel said. And one part, that's one part of the prize, is to go to the person who shall have done the most or the best work for fraternity between nations, or the abolition or reduction of standing armies, and for the holding and promotion of peace congresses. I think this prize given to the European Union fits perfectly those requirements, because it's given for the advancement of peace, and a united Europe has surely advanced peace, certainly over the last 60 years. It's given for reconciliation between nations, and it's also given for the advancement of democracy and human rights. Now, to understand not just what the economic achievement of Europe is, but the political and the cultural achievement of Europe is at the moment, just think back a little bit to the beginning of the last century. Between 1914 and 1918, you had a major war which destroyed practically most of Europe, with millions of people killed. Then, very quickly following that, in 1939 and 1945, you had another major world war with the same massive destruction of human life, the displacement of persons, also the persecution of minorities and particular cultures. So you get pogroms, you get the Holocaust, you get continuing genocides. So it's against that background that you begin to see both the necessity but also the huge achievement of a united Europe. It's not just, again, a nice political arrangement. There's a huge amount to be done in overcoming the mutual suspicion between nations, the contradictory and conflicting histories between nations. All of that is work that has to be done. So it begins in a very modest way in 1951 with an agreement to have, not to start on the big picture, but to start with what is achievable and what is practical in the Iron and Steel Union. And that consists merely of 6 countries: France, Belgium, West Germany, because remember you have the Iron Curtain, Luxembourg, the Netherlands, and Italy. Then you begin to get major growth happening. In the 1970s up to the 1990s, you get the addition of the United Kingdom, Denmark, Ireland, Portugal, and Spain. And then by this stage, of course, you also get East Germany because you have German reunification as a result of the end of the Soviet Union. The massive explosion comes in our own century between 2004 and 2007. There you have a huge number of Eastern European countries joining. Poland, the Czech Republic, Hungary, Slovenia, Lithuania, Slovakia, and Latvia, and then 2 little small places, Cyprus and Malta. So what does that actually mean? What has happened there to overcome this extraordinary, broken, painful, and fractured history of Europe, not just through 2 major world wars, but also through the Iron Curtain and the conflict of ideologies that goes on behind that? So it's not just about economic benefits. It's about social and human benefits. It's about the guarantee of rights for all the citizens of Europe. It's about the guarantee of a democratic politics for all of Europe. And so this allows for a deeper and more stable sort of security. At the moment, you have the total European Union consists of about 500 million people and accounts for about 20% of the world's GDP. Of course, there still remain tensions and struggles, because you can't simply bring together all of those nations with their unique individuality and their histories and suddenly expect it all to come together in a beautifully peaceful way. You wouldn't expect that. But what you do get is you get an extraordinary emergence of a different sort of, I think, a deeper and more mature notion of peace. Sometimes peace is thought of classically as an absence of conflict. But there can be a legitimate and productive conflict between ideas, between different political systems, as we're experiencing at the moment in the middle of the election. What really matters here is that that conflict is ordered through democratic and political processes to the search for the increase of a lasting common good for all peoples and all nations. In other words, conflict can take place in an ordered and productive way through political and democratic means. That's the major achievement of a united Europe. As well as that, you get the extension of human rights to people that, and particular to minorities and to cultures that have a precarious hold within different nations. So I think there is a huge advantage to what has been achieved. And just very briefly and in conclusion, how did it all happen? It didn't just happen out of the necessities, out of the pain and the conflict. It happened because you had some people who had the vision to make it happen. And part of the founding fathers of the European Union, Jean Monnet, Robert Schuman, Paul-Émile Spaak, Alcide de Gasperi, and Konrad Adenauer, are interesting figures in themselves. They weren't just very astute politicians, major players in their own countries. They were also, in the majority of them, very devout Catholics. They were inspired by Catholic social teaching. And it was that inspiration that made them want to heal Europe and achieve the benefits of a collective Europe for all its peoples. So, is the EU a worthy recipient of the Nobel Prize? Well, if the EU isn't, I'd like to know what really is. Because we've had 60 years of peace between widely varying nations with completely different histories. We've had the advancement of human rights. We've had the social expansion of welfare for all European citizens. But the prize isn't given for just what has been achieved. It's also given what still remains to be achieved. Because, as I said, peace is much more than the absence of war. It's the really deep security, not only of material and personal goods, but also of the spiritual goods, that we need what I would call a genuine ontological security. And that rests upon the comprehensive vision of what it is to be a human person. That we still have to achieve. Those founding fathers would have understood well the Jesuit-inspired motto of this university: change the world from here. They and their successors did. Thank you.
Thank you very much. That was very delightful. We now move to chemistry. And here we have John Cobley, who's a professor in biochemistry. He received his PhD from the University of Bristol. He currently teaches courses in biochemistry and in evolution. His research is on microbes and in particular their energetics and metabolism. For example, with funding from the National Science Foundation, he developed genetic techniques and used them to study how photosynthetic bacteria adjust to changes in the color and brightness of available light. I'd like to see that actually someday. Would you please welcome John Cobley?
So, I'm covering the chemistry prize, and what I'm going to be describing will seem like biology. The real reason for that is that science is progressing to such an extent that biological systems are becoming much more amenable to chemical analysis. And really, life is being understood as a chemical process. So basically, this is a brief extract of the declaration of why they received the award. Groundbreaking discoveries reveal the inner workings of an important family of receptors. So what are the key ideas here? The key idea are cells, receptors sensing an environment, and inner workings. So these are the two individuals who shared the prize. What's the point of putting up this slide? One is simply to state that they are both born in the US and educated in the US, and the other to point out that they are about 15 years apart in age. Lefkowitz actually took Kobilka as a postgraduate student after he had obtained his MD. So this really is the professor, this is the student, and they have subsequently shared the prize. So to get into this, how can I really approach it? This is a famous painting by Edvard Munch. It's called The Scream. We're going to think about really, with a little bit of poetic license, what might be happening to this individual. If this individual at the moment is experiencing some sort of terror, I'm sure it's really existential, but let's imagine that it's induced by these individuals at the back here. What sort of things are happening at the body? Here's a sort of description. Remember, you have in your body about 10 million cells. So we're going to think about cell response. And in cases of fight and flight, you perceive that you're in a dangerous situation. There's a nervous response from the brain that goes down to the adrenal gland. And the adrenal gland, which is here, releases adrenaline. And the adrenaline flows through your blood system, and it's a signal to your entire body that something pretty dramatic is going to happen, and you better really be prepared. And the body responds in many different ways. The heart starts beating faster, the heart pumps stronger. The liver, if you put adrenaline into the liver, glucose pours out of the liver. It's released from the liver to supply the body with immediate source of energy. Some muscles, their tone increases. Some muscles do the complete opposite. They really relax. There's a phrase that, excuse my expletive, but there's a phrase called scared shitless. And this phrase refers to the fact that while this hormone stimulates the heart and causes it to beat stronger, it causes the intestinal muscles to relax and you evacuate your bowels involuntarily. So adrenaline has a whole variety of responses. And what is this prize about? It's recognizing that what is the chemical understanding of what happens when that adrenaline is released into the blood and it goes to a cell in the body, could be a liver cell, could be an intestinal cell. What happens for that adrenaline to be a signal that effectively brings about a change inside that cell? Here's a brief shot of adrenaline, the molecule. It's very, very small. It has 3 oxygen atoms that are in red. It has, I think it's 9 carbon atoms and 1 blue atom, which is the nitrogen atom. So it's a very, very small molecule, and it's in the blood at very low levels. So this is briefly a little cartoon of a cell, how we're going to think about it. The cell is really like a bubble underwater. Just as a bubble in air has air in and out, the bubble underwater, the cell has liquid, water in and out. And these green little dots represent the adrenaline that's been released into the blood. And what we need to do is to have this adrenaline function as a signal to cause the cell to bring about some change. So it's now accepted that the adrenaline never gets into the cell. It interacts on the surface, and on the surface of the cell, you see what's called a receptor. And the receptor has a specific place where this molecule exactly fits. And when it fits, the molecule fits in there, and the adrenaline is present, the receptor changes its shape and interacts with a molecule inside the cell. And so really what we have is a chain of events starting with the signal outside, getting across this membrane that separates the in from the out, and this is the molecule that's going to carry the signal farther on. Okay, so Lefkowitz started to do this work, and he decided that since these receptors are there in very small amounts, he would make a little tiny molecule that would bind to the receptor and stick in it. And that molecule, that little molecule that was going to get into the receptor and stick, was radioactive. So once it had bound, he could follow where that receptor went, and he broke up the cells and tried to fractionate and separate out all the parts. And he identified a molecule that bound to adrenaline, and he was very excited, and he actually managed to figure out it's a protein, and proteins are made up of linear chains of like beads, like a string of beads, 20 different colors, possibilities. And at this stage, his student, who got the other Nobel Prize, who shared the Nobel Prize with him, comes on the scene, and he isolates the gene for this particular receptor. And what they find is if they line up the sequence of the protein, these are all the beads, it's similar in a way to the protein that senses light in your eye. And this is the protein that senses light in your eye. So there are 7 regions along this protein that have a special oily character. And it was known that the reason that these regions are oily is that the protein exists not as a straight line, but it doubles back on itself. And that's how it actually gets situated into the membrane. So at this stage, Lefkowitz describes this as the eureka moment because he realizes that there are 2 totally different proteins. The protein in your eye that's receiving a signal, the protein in your liver that's receiving the adrenaline signal, and they're very similar. And it turns out that there are in the human body about 800 different receptors, all have the same structure. They all sit in the membrane and they zigzag back across the membrane 7 times. And what are all these different receptors doing? There are receptors for taste. Instead of a hormone binding to the receptor, in this case, in the taste, there are 3 or 4 or 5 different types of molecule that bind to the tongue. When you chew food, the aromas, volatile gaseous molecules go up your nose and you sense them as smell. There are hundreds of different receptors in the nose that bind specifically to different smells. Hearing involves the same process. So what they've really found is a whole family of molecules that all behave the same and have the ability to respond to a chemical outside the body and convey information inside the cell. Now, this is a complex one. You don't really need to see all of this, but this is what happens in your eye when you absorb light. Here's the molecule that's zigzagging back and forth across the membrane. This is the receptor that we're interested in. In the case of light, there's a little tiny molecule bound there all the time. And only when it absorbs light does it change shape. And when it changes shape, it interacts with another protein on the inside, as I referred to before, and it goes off to do another whole series of events to bring about changes. Now, all of these 1,800 receptors in the human body, they all have these 2 parts in common. A 7 transmembrane area here on the outside of the cell, and this what's referred to as a G protein. So the holy grail in biochemistry has been to want to know what happens when a receptor binds. When a receptor binds a ligand. So here's what happens when you smell a molecule. This is what happens in cells in your nose. Here's the odorant, whatever it is, might be some perfume of the person sitting next to you, binds to the receptor, and there's the G protein again. Now the question is, what happens when the small ligand, that's the molecule that binds, change shape, and this then becomes interreacting and interreact. So these two molecules have to get together, and we want to know really what is the details here. And that is what this Nobel Prize is about, figuring out exactly what this detail is. Now, why is this so important? It's really important because half of marketed pharmaceutical medications are involved in binding to receptors and altering this interreaction. It is a big, big deal. So people want to know exactly how it works to be able to have the tools to make better pharmaceuticals. So what this really shows here is a membrane. Here is the receptor for the adrenaline. Here's the adrenaline binding. This was known, it's very hard to describe how these detailed structures are figured out. It involves purifying the molecule and then letting them form little crystals. And these crystals can be exposed to X-rays, and the X-rays are bent, and you get a photographic plate with this pattern of spots. And it's very sophisticated mathematics, but from that pattern of spots, it's possible to figure out this incredibly detailed structure. So what this particular slide shows is that when the little molecule binds, there's a change of shape here. And if we were to look now from inside the cell at the surface of the membrane, we would see this. You can see there's this vast dark area here that is produced by a change induced by this little tiny adrenaline-like molecule. And it's this dark area that is very oily and lipid-like that the second protein binds to. Now this is the shot that the Nobel Prize is really given for. In this particular shot, these two molecular complexes are caught with their pants down. They are caught in some molecular conjugal act. Here's the receptor. Here is a little molecule, that's the adrenaline-like molecule, and this is the G protein. This enables, this is only a cartoon. There is considerably more detail that's in this interreaction than can be just shown in this slide. But this really was what people wanted to know. How exactly do they interact? How, with knowledge of this, can we manipulate these interreactions in individuals that have serious problems, medical problems with these sensors? I mean, insulin itself doesn't bind to a receptor like this, but there are all sorts of problems with diabetes when people cannot sense insulin. There are equally other problems where individuals can't sense other molecules. The thing that I wanted to do in conclusion, that I can't really do from here, I wanted to be able to show for you how you can use graphics to illustrate and actually take these structures and manipulate them, rotate them, hone right in on certain parts, make measurements on these crystal structures. But the trouble is, I can't use the computer here. So I worked out this presentation to show it, but my computer is up there with the program in and I can't manipulate it. But this is an example of, this is a detailed description of the molecule that I last showed you. This green one is the receptor, and this complex of 3 different proteins, the blue, the red, and the brown, is the G protein that is going to carry the information away. And these can be rotated. And if you look here, we're now looking down onto the molecule as you would look if you look down onto the surface of the cell. Here is the ligand, the adrenaline bound, and that has caused the molecule to change down here so that this G protein can interact. So it's very sophisticated science. It's really chemistry applied to biology, but all this analytical technique is really physics. So this for me is shock and awe. Thank you very much.
Thank you very much. Well, you're very lucky to be a student at USF, I can tell you that. And our next speaker illustrates that even more. It turns out our next speaker is an expert in this area of literature. And our next speaker for the Literature Nobel Prize is Stephen Roddy, who's a professor in the Modern and Classical Language Department. He received his PhD in East Asian Studies from Princeton University. He teaches Japanese and Chinese literature, culture, and language. His research examines the influence of Chinese fiction on late Tokugawa writers and of Meiji period thinkers on essayists of the late Qing. I don't know what that's about, but it sounds fascinating. Please welcome Stephen Roddy.
So that was Qing, by the way, but that was a good try. So I'm talking tonight about the laureate in literature. His real name is Guan Moye in Chinese. But if you know Chinese, or if you know anything about Chinese, you know that it actually is a language with characters that are somewhat phonetic, but not exactly phonetic. It's a long story about exactly how to explain what they are, but what he's done is he's taken the middle character, he has a 3-character name, if you look at the middle, and broken it up into 2 pieces, and that has become his pen name, which is Moye. It literally means don't speak. So he obviously hasn't taken that to heart. He's done a lot of writing over the last 20 or 30 years, many volumes in Chinese, quite a few of which have been now translated into English. Now, probably most of you know that China has a very long history. Its recorded history is about 3,000 years old. And the beginnings of the Chinese written language actually go back even further, about 3,500 years. It's not quite as old as Egyptian hieroglyphics, but almost. The amazing thing, though, about Chinese is that literature written almost 3,000 years ago can actually be read by a Chinese today with a little bit of help, a few notes, an annotated edition. But it's actually quite possible for an educated Chinese person to read poetry written almost 3,000 years ago. And very few people can do that now. Perhaps if you've studied ancient Greek, you can. But very few Americans can read poetry written more than a few centuries ago without a lot of help. So I thought I'd talk a little bit about... Can I get this? Am I doing it right? Now I have a problem. Here we go. So I wanted to give a history of the Nobel laureates from China. There are actually some laureates in other fields besides the humanities or literature and peace. I believe there were a couple in physics. I don't know all of them. But Chinese who've immigrated to the West, in particular America, Europe, have won prizes in various fields of science. I don't know. I can't speak about economics. But the first one in Chinese literature, or literature won by a Chinese man writing in Chinese, was Gao Xingjian in the year 2000. After that, a few years later, about 10 years later, Liu Xiaobo, a Chinese man, won the prize not in literature but in peace. And he's actually a writer, not so much of literature per se, but of literary criticism. He is considered a literary critic and also an activist. So both of these men actually were associated with a very important event in China that happened a little over 20 years ago. In 1989, you may remember, although you may not have been alive then, some of you, that there was a massive wave of unrest in China, and it all came together in Tiananmen Square in the spring of 1989. It turned into a violent protest, or rather not violent on the part of the protesters so much, but the government's reaction was fairly violent, and a number of people were killed both in Beijing and around the country. So these two men were involved indirectly in those protests. They actually weren't, neither of them were in the square itself, but both of them actually became known for their activism afterwards. Gao had actually already moved to France before then. He went back to China during the protests and then left afterwards, moved back to France, and became very active in political protests against the repression of Tiananmen, but also other causes related to China during that period. Liu also returned to China actually during the protests themselves, and became very active in various movements there related to democratic change. Eventually, he was arrested in 2009 and sentenced to, I believe it was 15 years in prison. So these are the two recipients of Nobel Prizes, one of them actually now a French citizen, the other one in China still, but sitting in a jail in Beijing. He wasn't allowed to go to Norway in 2010 to receive his award. And, oops. I'm going to go forward here. I guess I'm not very talented. Can I move to the next slide? Hmm? That way. Over there. It worked. Oh, great. Okay. So we don't know what Moyen will say when he goes to Oslo in a couple of months, but we do know what Gao Xingjian said about 10 years ago when he won the prize. And Gao is known, as I've said, as a figure in dissident circles, very much engaged in various political movements that are trying to change China from outside mostly, but also from within China as well. When he went to Oslo to deliver his address, he talked very much about his work as a form of social and intellectual protest. And that's probably the main reason why he received the prize. I don't know if many of you have followed the history of the prize in literature over the years, but many of the writers are kind of forgettable as novelists or poets, but they were often associated with particular political movements or were kind of dissidents and were repressed in their own countries. So the prize has often been used as a way to kind of recognize people who were working for what Norwegians and Swedes and many others thought to be worthy of support from the outside. Aung San Suu Kyi, of course, being a recent example as well for the Peace Prize. So this history of the Nobel Prize in China has elicited a somewhat mixed reaction from the Chinese street. Of course, many people in China today probably would wish for and hope for a more democratic, a more egalitarian, a more open system of government and expression to come about. But many people were not terribly happy to see Gao Xingjian receive the prize in 2000. Most people hadn't read him. Very few people think he's a very good writer at all. So his prize was not well received in China. Liu Xiaobo is also kind of a mixed bag, although he's very well respected by many people, both in China and outside. He also received support from various organizations in this country that have kind of put him in a bit of a funny light in China, because there's a very strong sense that in China, political change must come from within, and it shouldn't be imposed from without. You might say, in some cases, that's a consent that's been manufactured or at least manipulated by governmental propaganda. But it's still a very strong feeling among a lot of Chinese that they don't want outside forces, whether governmental or private, to be influencing their politics in really any way, shape, or form. So these prizes have been pretty controversial within China. So, what about our 2012 recipient? I think people feel very differently about that. He is an author who lives in China today, has never spent very much time abroad, and he's identified with a particular place in China because almost all of his work is set there. And that place is a province which is near the city of Qingdao. Many of you may know Qingdao because of its beer. It is the best beer in China. I think so too. But his area is actually in the interior. It's called Gaomixian, or Gaomi County. It's a rural area, has never been famous for much of anything. So probably Mo Yan is the most famous resident of Gaomixian, past and present, and maybe future as well. There we go. So what's he famous for? He's not the only writer today that's popular and well-known in China. But he's well-known as for representing a kind of tendency among modern Chinese writers, which is to represent the past and particularly the traditional past, the past of farmers, of traditional scholars even, of people who lived in a society that had not been terribly affected by modernization. So there are a lot of writers today in China who have picked up on this trend of writing about traditional society, and he's one of them. He also uses a lot of traditional literature in his works, and he draws on the models of literature that come from his own native area. So there are two novels that some of you may have heard about because they've been translated into English. Jinping Mei is one, Shui Hu Chuan is another. There are two novels that are considered classics of traditional literature, and they're both set in an area not far from Mo Yan's native place in Shandong province. He's also very interested in tracing the history of China of really the recent 3 or 4 generations. So many of his works, in fact, most of his works, deal with the China of the Revolution, the China of the 20th century, when it changed really from being a traditional agrarian and imperial order to a revolutionary one, dominated first by communism after 1949, and then, of course, more recently by very strong and vibrant, you might say, or perhaps overpowering capitalism. Okay. So there are other writers in China. He's not the only one that's important. Many people are very fond of Yu Hua or Wang Anyi, and there are several other names that could probably be considered rivals for Mo Yan, with Mo Yan in one form or another. Oops. Sorry. How do I go backwards?
Flip it over. And then look at the top. Oh, upside down. Sorry. You just flip this. That's forward.
Oh, I'm sorry. I was using, okay. Thank you. Oh, okay. It's up there. Oops. Let's see. There we go. Oh, so I get it. Sorry. So the Nobel Prize Committee has basically pointed out certain features of his writing that they particularly appreciated. And most of all, it's perhaps this kind of combination of realism and a kind of fantasy, or surrealism is another way to describe it, or the grotesque is another way. Some people mention him as being a spiritual heir of Garcia Marquez, other writers, Faulkner even, people who combine a kind of a very strong interest in the local culture of a particular place with fantasy, spiritual, or otherwise. So I wanted to actually just read the first lines of probably his most recent work that has been translated into English that is considered in some ways his masterwork. This is an interesting story, and it's set in the latter half of the 20th century. So it begins with the following lines. My story begins on January 1st, 1950. In the 2 years prior to that, I suffered cruel torture, such as no man can imagine, in the bowels of hell. Every time I was brought before the court, I proclaimed my innocence in solemn and moving, sad and miserable tones that penetrated every crevice of Lord Yama's audience hall and rebounded in layered echoes. Not a word of repentance escaped my lips, though I was tortured cruelly, for which I gained the reputation of an Iron Man. I know I earned the unspoken respect of many of Yama's underworld attendants, but I also know that Lord Yama was sick and tired of me. So to force me to admit defeat, they subjected me to the most sinister form of torture hell had to offer. They flung me into a vat of boiling oil, in which I tumbled and turned and sizzled like a fried chicken for about an hour. Words cannot do justice to the agony I experienced until an attendant speared me with a trident and, holding me high, carried me up to the palace steps. He was joined by another attendant, one on either side, who screeched like vampire bats as scalding oil dripped from my body onto the audience hall steps, where it sputtered and produced puffs of yellow smoke. With care, they deposited me on a stone slab at the foot of the throne and then bowed deeply. Great Lord, he has been fried. Having been fried to a crisp, I knew that even a light tap would turn me to charred slivers. Then, from high in the hall above me, somewhere in the brilliant candlelight, I heard a mocking question. Shima now, is more rioting in your plans? And he goes on to say, yes. And Yama, the king of hell, goes on to torture him further. So this is the opening lines of a book about 4 incarnations. Basically the story of a man who lived as a very cruel, very terrible landlord in the period before the revolution. He's sent to his death right at the beginning of the communist era. And then he's reborn as 4 different animals, a donkey, a pig, an ox, and a monkey. And so the story pretty much takes his point of view as he describes China evolving from a revolutionary society in 1950 to roughly 2000 or 2005, when this area, his hometown, Gaomishen, has been turned into kind of a theme park for capitalist investors from the West, from Japan, from Taiwan, and his whole hometown becomes a kind of bizarre sort of echo of various different stages of Chinese history, ranging from the 16th century, because the name of the hero comes from the hero of a book written in the 16th century, all the way to echoes of Chairman Mao and the post-1976 reforms in China. So it's a very interesting book. I recommend it highly. But he has written a number of other novels, including Red Sorghum, also Wide Hips and Big Breasts, a number of books that you might be interested in taking a look at. Red Sorghum is probably the easiest to read because you can also watch the movie. And the movie was made, it's getting a little dated now, about 15 years ago, starring Gong Li, who for many people is the most famous Chinese actress. So there are a lot of ways that in which you can find out more about Mo Yan through his books, through the films that have been based on his books, and you can find a lot of interviews with him as well online. He's increasingly being translated into English. I think this is an important prize. Mo Yan is certainly a deserving author, and obviously he's going to be very prominent in China now. In fact, I just found out that they're turning Red Sorghum, his first and sort of biggest hit in the 90s, into a theme park in his hometown. So indeed, he's going to be quite a celebrity in China for the rest of his life and probably long after that. Thank you very much.
Okay, thank Thank you very much. Thanks to all our speakers. That was wonderful. We can open it up for questions right now. Does anyone, if you have a question, just raise your hand, and then we'll figure out a way to do this. Any questions? Everything was answered, is what you're saying. Okay, well, there is a question.
I just have a question about Schrodinger's cat. And I was fascinated when you said, there are these two simultaneous states. But then you used the word choice. And I didn't quite understand how, if you're in two simultaneous states, you can actually have a choice.
Thanks. That's a perfect question, and I don't know how to answer it. And the best way I can answer it is that no actual choice is made. Nature doesn't choose. What Serge Haroche showed in his measurements is that the superposition of states, meaning that the cat is both alive and dead at the same time, eventually seems as though the probability for being in one or the other dominates. So the probability for being alive, let's have an alive cat. It'll make me sad, but maybe some of you happy. The cat lives, and it's because this careful superposition of states becomes completely mixed up with the environment. The rest of the universe interacts with this carefully prepared state, and the universe that we live in seems to be a universe where the cat lives. Although what has really happened is that the superposition has become blended with the rest of the universe. And what's cool about the prize is that they were able to keep these quantum systems from blending with the universe for long enough to watch that happen. But I'd be happy to talk with you more anytime. Thanks.
Any more questions? Anyone else? You've got these experts here for a couple more minutes.
How soon will we be able to see the results of this research in the areas of stem cells replacement therapy for things like Parkinson's disease?
So that's a great question, and I mean the reality is it's happening now. I mean we're doing it for some diseases. We haven't done it for Parkinson's disease necessarily, but there's other diseases that we're actually going in and doing stem cell treatments for, things that we can manipulate a single cell to actually change those cells. We do it a lot with several cancers. It can be done with leukemia now, so we can actually take pluripotent cells to replace those dying bone marrow cells. But as for Parkinson's disease, that's a tough one because neurons are a little different in the human body. They're the only cells that can't divide. So if we can take them and pull them back into a pluripotent state and then push them back in, make them take that trajectory of a non-dividing cell, it's a very, very tricky game. So it hasn't happened for the neurological diseases at this point, but we're beginning to actually see that stem cell therapies are working for several diseases that can be treated by reintroducing some cells to replace those damaged or dying cells. The biggest issue, and if you look at anything by talking about the prize, is the fact that this is basically a research tool at this point. We're not using it as a therapeutic tool. So it's mostly allowing us to understand the diseases more so than treat the diseases at this point. So is it going to happen tomorrow? Probably not. But is it going to happen in our lifetime? Probably so. So that's my best guess.
Thank you. Anyone else? Is that it? Okay, well, thanks again for our great speaker. Oh, we have a question. Okay, great.
Mine was for economics.
Oh, she's not here. Okay.
I want to ask a question about the literature. And as we all know, the famous Japanese writer name is Murakami Haruki. And can you show me about he and compare with the Mo Yan and who hardly missed the Nobel Prize this time? And I think so. And Murakami Haruki will win the next time. What do you think?
Did you get that? Can you repeat it as well?
Sure. So there's a Japanese writer by the name of Murakami Haruki who was rumored to be on the short list this time. I think he was nominated. And many people were disappointed when he didn't make it. So I think he will get it eventually. Perhaps the reason Moyen beat him out this time is that Murakami, for much of his career, has been considered a kind of very apolitical writer. So although his recent work has been more committed, you might say, or more, it has a more political flavor to it. He has actually spoken out quite strongly since the tsunami of 2011 about the need for Japan to reform politically and economically in this terrible situation they're in. I think it's because Moyen has really, although he's very middle of the road, his work is quite critical of many aspects of China today. As I said, this last work that I just described shows the transformation of a rural county from desperate poverty through the Mao years to this kind of explosion of economic growth that has resulted in a really a moral desert, you might say. And so Moyen's work, I think, was probably evaluated for its political relevance to China today. And I think Murakami probably lost mainly for that reason, but I think he'll get it eventually.
That's it? Any?
I have a question for biochemistry. I want to know how exactly the ligand induces the receptor to bind to the G protein. Like you said, it changed shape, but how does it know that it has to bind to the receptor?
The ligand that's in that work that created that crystal structure is not the natural adrenaline. It's a very tightly binding, and it binds even more tightly with adrenaline. But if you think about it, there are 7 of those helices. They are rod-like structures that cross the membrane. So it's like if you had a bunch of sticks and you squeeze the sticks together at the top, the other ends spray out. So the rods are attracted chemically to the ligand, and the bottom part sprays out, opening up an area where the G protein can bind. That's the best answer. We can talk in great detail, but that's the best description I can give.
Anyone else? That's it? We're done? Last chance. Sorry about economics, but she had to go teach a class. No one else? Okay, can we give another great round of applause for these great speakers? Thank you, Ben. Thanks very much for coming.